Hello, welcome to another edition of 5-Minute FRQ, where I take you through an old FRQ from the AP Bio exam in 5 minutes or less. As always, this information is freely available on the College Board's AP Bio website, so if you have any questions, you can look it up. Okay, this is question 7 on the 2019 AP exam. A researcher is studying patterns of gene expression in mice. The researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of messenger RNA from 6 genes. The data are shown in figure 1. And then the question presents me with this sort of chart type figure where on the left I have 6 different tissues from a mouse: liver, heart, brain, kidney, pancreas, and skeletal muscle. And on the top I have 6 different genes: gene E, F, G, H, I, and J. And the key tells me that the darker the box is filled in, the more messenger RNA those cells have. And what you have to know about this question is on gene expression, and you have to know that if a gene is being expressed, those messenger RNAs are going to be found in the cytoplasm. So part A, based on the data provided, identify the gene that is most likely to encode a protein that is an essential component of glycolysis. Provide reasoning to support your identification. So now this question throws out glycolysis. So this is putting multiple topics in one question, which is great. So glycolysis is a process it's pretty universal in mammals. How many of your cells are doing glycolysis? It's pretty much all of them. This question is a mouse, not a human, but you're both mammals. So the gene where the RNAs are found the most is gene G, all right, because all the boxes are filled. You got a 1 in 6 shot. So if you have no clue, at least name a gene. So the reasoning would be because all of those tissues do glycolysis. If you look at the rubric, gene G, the identification was worth 1 point. And the reasoning would be because gene G is expressed in all 6 tissues and they all do glycolysis. Part B, the researcher observed that tissues with a high level of gene H messenger RNA did not always have gene H protein. Provide reasoning to explain how tissues with high gene H messenger RNA can have no gene H protein. So part A was talking about transcription, how when you transcribe DNA into RNA, when the gene's turned on, you're going to have RNAs, right? Part B is talking about translation. And it's saying, how could you have the messenger RNAs, but not have the corresponding protein? And the process that turns RNAs into proteins or reads RNAs to build proteins is translation. So in a nutshell, if you don't have the proteins, it's because translation is not happening. So why might translation not be happening? So look at what the rubric took. So the messenger RNA is not being exported from the nucleus. So maybe you've made the RNAs, but they're not in the cytoplasm yet. So you're just not there yet. The second thing, gene H is not being translated. Again, so if translation is not happening, then you're not making the protein. It mentions RNA interference. You really don't have to explain why. You can just say it's not being translated. And the last point, post-transcriptional modifications. So oftentimes when a gene is transcribed, you have the introns being cut out, you have the editing, you have alternative splicing. Maybe those modifications are happening toward the messenger RNA that you're looking at is not the final transcript, which is being turned into the protein. Saying any one of those 3 things got you the point. Okay, hope that was helpful, and I'll see you guys next time.